In the last video, we looked at some of the exceptions to Mendel's rules. And we're going to continue looking at exceptions to Mendel's rules, starting with sex-linked traits. Sex-linked traits are traits that are controlled by genes that are on the sex chromosomes, usually the X chromosome. Autosomes are the non-sex chromosomes, not the X, not the Y, but everything else. Autosomal inheritance tends to follow more your traditional rules of inheritance. But the sex chromosomes, if they're on the X chromosome, because different sexes get different amounts of the X and the Y, that can affect how those traits are inherited. So this was first discovered back in 1910 by Thomas Hunt Morgan at Columbia University. He was breeding Drosophila. Drosophila has been a standard subject for inheritance, for genetics, because they reproduce quite well. They make lots of offspring. They have short generation time. They're easy to care for. And they've only got 4 pairs of chromosomes, so they're pretty easy to study. Fruit flies, like humans, 2 X's, female, X and Y, male. This is a human karyotype, chromosomes 1 to 22. Both pairs look very much like each other structurally. Those are your autosomes. X and Y, those are your sex chromosomes. So this individual would be male because they have an X and a Y. So while he was doing this, he discovered some white-eyed male fruit flies. So he decided these were a mutant, whereas the red are the wild type or the normal eye color. So you take a true breeding red-eyed female and a true breeding white-eyed male, and like in a typical Mendel cross, you would cross them, and what we see is all the offspring, both male and female, have red eyes. That's pretty much what we would expect. Red eye color is dominant to white eye color in this case. But when we did the F1 crosses to produce the F2 generation, this is where things got interesting. All the females, red eyes. But in the males, 50% had red eyes and 50% had white eyes. But there were no white-eyed females at all in the F2 generation. So something clearly different was going on here. So let's look at the Punnett squares here. So here's our F1 cross. Cross these guys to produce the F2 generation. This is what we would expect. A 3-to-1 ratio. It's not what we got. Instead, because 2 X chromosomes typically develop as a female, there's some redundancy, very much like an autosome. Because males only have 1 X, you only need 1 copy of each X gene. So when we look at the Punnett square with our sexes here, 50% chance of being female, 50% chance of being male. So let's look at this a different way. Let's include the sex chromosomes in our Punnett square. And this is what we got. Again, all the offspring had red eyes because everyone had at least 1 big R gene. Across the parents that are the F1 flies, because the male has a big R, it's always going to donate a big R to any female offspring because that's all it can donate. But mom has 1 big R and 1 little r, so 50% of her male offspring are going to receive a big R, and 50% of her are going to receive a little r and be white-eyed. So again, if we look at our Punnett square, you can see, and this explains the results that we got when we did this. There are genes on the Y chromosome. The main gene we usually think about with the Y chromosome is SRY, which is what makes a person male. But there are some other genes there. but they all have to do with maleness. That's all the Y chromosome does, is it makes a person male, because if there are essential genes in the Y chromosome, the females would be missing them. But the X chromosome does a lot more than determine sex. It has genes for needed things, like blood clot factors, color vision, and so on in humans. So we call these sex-linked traits or X-linked traits, and there are about 60 different diseases that we can trace back to genes on the X chromosome. And you can see them on the gene map here. The Y chromosome only has about 30 genes. And this is kind of a joke here, as you can see. One thing that's interesting is that hemophilia, or at least factor VIII hemophilia, which clotting factor VIII is missing, we've actually, because of it occurred in one of the royal families, we can actually determine where that first mutation occurred. It was in one of Queen Victoria's offspring. So here's the pedigree for Queen Victoria's family. You can see Queen Victoria is a carrier. She's half filled in. She's heterozygous for hemophilia. Because she has one good copy of the gene, though, she's okay. But Leopold, you can see, he's got hemophilia because he only has one gene for blood clotting. So you can see in the pedigree, all the affected individuals are male. There are no affected females. There are females that are carriers, but none of the female offspring had factor VIII hemophilia. So how can we explain this again? We can go back to our Punnett square. Carrier female, normal male. You get all normal females. You get 50% males are normal and 50% have hemophilia. Now, because of this fact that humans, because there's two X chromosomes and we only need one copy of them, in order to avoid overproducing the proteins on the X chromosome, in females, one of the X chromosomes becomes inactive. It becomes very, very highly condensed in something we call a bar body. But it's random which one, whether it's the X chromosome for mom or the X chromosome for dad. So if you're heterozygous for a trait, you're going to have some cells that have the dominant trait and some cells that end up with the recessive trait. And so we get what we call genetic mosaics. Calico cats are a great example of this. In some parts of their body, they're going to produce the brown fur color versus the black fur color, or the orange if you want to call it that way. Because when the cells formed in the early embryo state, in some cases, the X chromosome from dad got inactivated. In some cases, the X chromosome from mom gets inactivated. And so you have, in certain areas of the body, different colors produced. Almost all calico cats are going to be male. There is a case where you can have an XXY male, but that's the only way you could get a male that was, or a female that was calico. Pattern baldness is what we call a sex influence trait. Sex influence traits are inherited differently in males and females under the influence of the male and female sex hormone. So in males, because we have more testosterone, pattern baldness is a dominant trait, but females, it's a recessive trait. That's why we see more pattern baldness in males than we do in females. The environment can affect the expression of a gene. So there's cases where the environment and the genes determine phenotype. Human skin color. The amount of melanin you can produce is genetically determined, but you produce your maximum amount of melanin when you're exposed to UV light, especially if you're lighter skinned. Coat color in Arctic foxes is influenced by heat-sensitive genes. So when it's warm and there's not any snow, they produce more browns and black fur color, so they blend in with their environment better. When it's cold and there's lots of snow, they don't produce the color. They're white. They blend in with the snow. Hydrangea is a type of flower that's influenced by the pH of the soil. In acidic soils, it produces one color flower. In more basic soil, produces a different color flower.